So we have seen, or hopefully I put you in the idea of how genetics has advanced. I told you how important are mother organisms. And this second part is actually the importance of this variation. Yeah, make sure that you sign the register, okay? So the importance to have the variation in genetics and for genetic analysis, okay? So for that, I'm going to go quickly through what are mutations and how they are and where they are coming from, and then different ways to do mutations in plants, okay, that they are actually used these days. So as you know, mutations are just changing genetic sequence. Can be actually spontaneous, or it can be induced. Different factors, as we will see later on, can induce different mutations, okay? And spontaneous, it can be just inside the cell by different metabolisms. So where do you think it will be one of the main sources of mutations in your replication errors? Yeah? Another one? Oxidation and reagents that they could be actually involved in oxidative oxidation. So there's quite a lot of different things that actually inside the cell could produce these damages. We will see more in a second too. But yeah, so there are different things that they could be just spontaneous. They can be classified in different ways. And I just put here different examples. The most normal one is if it's a dominant or recessive, so depending how the phenotype comes out in the segregation. But you could have another one that we use quite a lot is the loss of function or null mutation. So this is basically the gene is so badly damaged that it won't produce any protein, okay, at any level, and it will have a full knockout effect. Some of them can gain function, so there are weird things that they happen. There are lethal mutations that they are quite difficult to analyze, as you can understand. So if it's lethal, you don't have anything to analyze the phenotype or the genetic changes. They can be harmful, beneficial, neutral, so you got lots of things. The pattern of inheritance is quite important because as humans, plants are diploid species. So we will be talking about homozygote mutation, when you have the mutation in both of copies of the genes, so your mom and your dad, and heterozygous when it's only one of them that contains the allele, mutated allele. Sorry. So, as I said, endogenous damage, so normally replication errors are the key thing, but also oxidative radicals are actually quite a big part of DNA errors. Exogenous damages from radiation to chemicals, even viruses can produce this. So quite a lot of different damages. Normally, and I put always this because it's quite scary, we have 1 million lesions on our DNA per cell per day. So it's quite a massive amount. The good thing is that normally it goes to DNA repair, and there are different pathways that they repair. So if it's a single strand damages, we got all these pathways. If it's double strand break, so it breaks the two strands of the DNA, basically you have different pathways. And we will see more about that. I don't want you to memorize all this, okay? I won't ask you too much about that. The key thing that you are going to see that they are going to be very important for the workshop is how these double strand breaks are repaired, okay? And how I use homologous recombination and homologous enjoining. And that will be linked what we will see with the workshop, okay? The workshop is about genome targeting, I didn't say yet. So, and this is something that is quite fashionable these days, so I think that will be quite important. But the rest, don't worry too much. So, basically, if they are not repaired, it will go to senescence. Cells start to accumulate errors and don't produce the proper things that they have to do. So they get old, the cell dies, or it actually develops to cancer. Okay. So quite important to be able to repair all these things. And in a way, there is a correlation of what the damaging agents are, what the errors are produced, and how they have to be repaired. Okay? Because normally all the repair pathways require to localize the error first to be able to solve it. And again, different errors actually or damages can actually produce different pathways to repair. And for example, for double-strand breaks, ionizing radiation and some anti-tumor agents can produce these double-strand breaks that they will have to repair, okay, between different pathways. And we will see more about these bits in our workshop, okay? Because, as I say, homologous recombination and non-homologous joining will come out as two of the pathways that they use for genome targeting. So when I'm talking about genome targeting, you're following me, yeah? CRISPR-Cas, magic word. So we will talk more about that, okay? So mutants. So this is just a slide just to show different mutants in Arabidopsis, of how the normal leaves in the rosette start growing and how different mutations actually can come from different genes, and actually you can form a very nice linkage of the metabolism of the leaves architecture by different mutations and how they are connected with each other, okay? So this is basically what you do in any other organism. So in plants, it's no difference. This is just to show you the differences in between forward and reverse genetics that I'm pretty sure you have seen with Matthias, didn't you? So basically, I always put this picture because it's actually quite visible. So forward genetics goes from the phenotype that you see. So you see a mutant that the flower doesn't look normal, and then you try to analyze what actually is changed in the genome. And the gene location that you find that there is a change or a mutation, that will explain why it is like that. Reverse genetics is a different way. You have one gene, and you want to actually ask the question, what is this gene important for? So you produce a mutation in the gene, and then you check the phenotype, okay? And this is what we actually call functional genomics. We got lots of genomic data, a lot of sequences, but we don't know what actually most of the genes are doing for and what they are important for. So this is a very good way to analyze all that. So, for the forward genetics, basically, the problem is that you are looking for a mutation, and you want to select that mutation. So in that case, it was a flower mutant. So you just grow lots of different mutants, plants, and then check for a mutation that is interesting for you, okay? So that's kind of, as I was talking previously, the selection of what you want to. In plant breeders will be a better yield or better things for the plant. For a mutant, that will be that you want to analyze just for whatever phenotype you are interested. One of the things or steps that they are quite important is to classify the mutant, to know if it's dominant or recessive. So basically do crosses with the wild type and see how it segregates. So it's basically a Mendelian system. If it's one gene that is affecting that mutation, or it might be several genes. So all these things are quite important. If it's one gene, you will be able to map the mutant to a location. So again, doing crosses and actually do a genetic map or linkage map. You can use, in some species, there are several lines that they have deletions or duplications, so you could actually analyze in which chromosome is, or in which part of the chromosome are. These days, you can just sequence, okay? So depending on how much funding you have or not, okay? Once you know which gene is and what type of mutation is and where the mutation is, even the sequence of the mutation, the first thing you have to do, and in plants, in the plant community is very important, is what we call the complementation test. Why this is important? Anyone?
Because you've got to show that what you knocked out is actually causing the effect.
So basically, if you put a gene that is the wild-type gene, you will be able to reverse. In humans, put it this way, in humans it's what we call gene therapy. So in plants, we do that all the time, once we want to demonstrate a gene is mutated, okay? So we just put the right gene and see if actually this mutant phenotype gets reverted into the wild type. Yeah? So in that respect, we are a little more advanced than humans, so research. So reverse genetics. So we just, as I said, you start with a gene. You ask yourself the question, is this gene important? What the hell is this gene doing? And basically, this is something that normally, if you are in a research lab, some major discovery happen in another organism. Okay, so in our case, yeast is mostly used to actually study meiosis. So suddenly someone published something in yeast saying this gene is very important, or this mutation is very important in this gene to actually control this part of the meiotic recombination. So basically, we check if the plant has it. We found a putative gene that it could be coding for that, and trying to do mutations to analyze if we have a similar defect or not. And most of the cases is correct. In other cases, we found different things because they are different organisms. But this is actually driving, reverse genetics is driving most of the genetic analysis these days, and functional genomics. So the first thing we have to do is actually produce a mutant, okay? And how we are going to get this mutagenesis working in a specific sequence or in a specific gene. So in plants, we are going to see a few examples, but you can use with different chemicals like EMS. We will see in a second a very nice technique that it was for the first time done in plants. You can use radiation, so gamma rays is a good source. In plants, it's quite nice because we can use insertional mutagenesis, or tDNA. When I'm talking about tDNA, everyone knows something about that? So it's a plasmid that gets integrated randomly in the genome, and it comes from a bacteria, okay? And I will explain more about that, so don't worry. The only problem with all these mutations is they are random, okay? You cannot say, I want a mutation in this gene in this position using these techniques, okay? They will produce mutations on the genome, but they will be random, okay? So quite a good important bit of actually producing mutants is the screening, okay? So quite a lot of people produce tens of thousands of mutants, and then they check where in the genome these mutations have occurred, okay? And this is the quite labor part, because you have to check all the sequence where these changes have happened. Mostly use PCR, restriction enzymes, sequencing, and we will see a few examples of that. But basically, you have to find where all these mutations have happened, and then find some that they have happened in your gene, okay? So as you can see, it's not that simple. Obviously, I didn't include here the genome targeting, that these days is changing all this. So we could actually target with CRISPR-Cas a mutation in a specific gene, okay? This is what we'll see in the workshop. But this technique, TILIN technique, so it's a targeted induction of local lesions in genomes, has been massively used in zebrafish, for example, and in mouse, but actually was pioneered in plants for the first time. And it's combining two things, combining chemical mutation, that it will produce random places of mutations, with a modern high-throughput genomic screening, okay? And it could be genomic sequences, or it could be different things, as we will see in a second. So try to follow me. So they use, the chemical mostly use ESMS, or ethylmethane sulfonate. And it basically is used because it induces a single nucleotide base exchange. Okay? A point mutation is what we call. And obviously, you can use different concentrations depending on the species or how many mutations you want to include. So normally, in the lines that have been analyzed until now, the average is 1.5 mutation per seed that they have been treated. Okay? Some of them will not have any mutations, some of them will have 2 mutations, and some of them will have 1. So it's a very good number to actually start working with that. Once you've been treated the seeds that they contain the embryo, remember that, basically you can put them on soil and grow them. And this is what we call the mutation generation 1, or M1. And from this one, depending on which species, I put self there because this is an example with Arabidopsis. You have self-cross, so the plant can cross with itself, okay? But if not, you cross it with the wild type, okay? And you obtain what we call the second generation of plants. And it's actually at this generation when you collect DNA, okay? You extract DNA from the plants and also keep the seeds of each individual plant properly labeled, okay? What happens next is that you pull all the DNA from these plants together, so you can put up to 100 plants or even 1,000 plants in each pool, so you can put them all together, as long as you know which plants are in each pool, okay? This is something that you will have to bear in mind. You can do pools with different plants from different ones, so you can put plant 1 here is always the same, but then all the rest of the wells could be from different plants containing, so you can do different experiments in different ways. But the key thing is that you are going to pull DNA from different plants in the same well, okay? And this is very important because we are going to use what we call the DNA mismatch cleavage enzymes, that they are going to be quite important. So basically you have DNA in each well that is coming from different plants. So what we will do is actually, because we are interested in our specific gene, so we know the sequence, we use primers that are specific for that gene to actually amplify that region of interest, okay? And you denature and renature several times all that PCR products, and then all these products can be cut with cell 1. So cell 1 is an endonuclease that recognizes mismatched DNA. So basically, you have a point mutation of one of the strands, and because of the naturing and renaturing of DNA, because you have DNA from different plants, you could have a hybrid with that it has renature of a plant that have one strand that it was mutant and a plant that it had a strand that it was wild type. Yeah. So if that happens, that will be a proper trigger for cell 1 to actually produce a cut. Okay. And as we will see later on, you could run in a gel and see these fragments, or you could actually use fluorescent probes or primers labeled with different dyes to actually see that. So it works really well. Cell 1 actually comes from celery. That's why the name. And then, obviously, once you know that in one of these pools you have something that it could be a mutation in your specific sequence that you are interested, you could actually come back to that pool, reduce the number of plants in that pool until you find the plant that you have. And because you have the seeds collected, then you can grow homozygous or heterozygote lines in the next generation. So this is, again, the example. You have the mutant population plant M2. You extract the DNA. This is an example of primers that they have been used with different color dyes, so fluorescent probes, to amplify your specific gene sequence. So normally you will have PCR products, as you can see that they are the wild type like, but for one of them will have a point mutation, and when you denature and renature by random, you will have some of these products actually have half a strand from one wild-type-like, and half of 1-point mutation. You cut with the restriction endonuclease enzyme, cell 1, and that will cut in this mismatch part of the hybrid DNA molecule. And then basically you just run on a gel, or even if you use fluorescent probes, it could be just because quenching, you could actually recognize which pool contains a fragment. As you can see, if it's the full fragment, it will have blue color because it's quenching the green color. And if it's cutted, then the blue and the red is different, so you will have, the green, I'm colorblind today, and the green will be visualized. So you could actually see quite nicely like that. These days, because of the cost of sequencing has gone so much down, quite a lot of experiments done in crop species actually is using sequencing. So straight away just sequence until they found point mutations. So good things of this, you have a very big coverage of the genome, so you can find mutants for lots of different parts of the genome, different genes. You can identify different alleles. Remember that EMS is doing a point mutation, so it's just a one nucleotide exchange. So you could have alleles that actually are not knocking down or knock out the gene, but actually alleles that they are doing different things. So you can have, for an hypomorphic, reducing basically the outcome of that gene, or hypermorphic in some cases where actually the gene works better for some strange reason. So, as I said, this is useful when knockouts or null mutants are actually lethal. So, in this case, if you put a null mutation and it doesn't go forward, it doesn't grow, then you cannot do any kind of analysis. But if you have one of these hypomorphic mutants, you could actually do some analysis on that and see, okay, it's not that good doing this or doing that. This actually doesn't produce any GM, so it's not controversial. As we will see with the tDNA, it's more controversial because it produces transgenic plants. But this is not transgenic. This is a chemical mutation, so it doesn't count as transgenic. And it's been massively used by plant breeders. So they are tilling lines for peanuts, soybean, barley, maize, sorghum. We use brassica, wheat, so it's quite a lot. And this is a very nice, actually I have to put it in the, I think I put the link, but if not, I put it. It's a very nice review. It's a little old, but it's a basic review for how tilling is working. So, tDNA insertion mutagenesis, okay? So this is coming from Agrobacterium tumefaciens, that is a bacteria that basically contains a plasmid that is called the TI plasmid, and this TI plasmid contains the tDNA sequence, okay? And this tDNA is the part of this plasmid that it will get inserted into the plants, okay? This is something that is being done as a bacteria to survive inside the plants, so they have some virulence genes in that tDNA that allow the bacteria to grow into the plant and basically hijack all the metabolism of the plant cell that they have been in to actually survive. But what quite early on in the 90s scientists did is take these virulence genes to be able to have Agrobacterium and actually the tDNA ready to do biotechnology, to be able to insert bits of DNA inside plant cells. Okay? One of the things that is quite handy is that it is called the tDNA as a binary vectors because they can grow in E. coli. So you can do all the growth similar to the cloning you do with anything else in any other organism. And then once you have it, all the constructs done, you just have to transfer them to Agrobacterium to be able to transfer them to the plant. Okay? So it's a kind of three-step thing. You use E. coli to produce your constructs, and then you pass it to Agrobacterium to be able to transfer to the As we will see too, it's very important to have selectable markers, and quite a lot of the controversy of the GM comes from that, okay? So it's something to highlight that we will see more next week, but basically in the vector, in the plasmid, you need a marker for the bacteria growth to know that you have the plasmid, and you need another marker for the plant to know which plant actually has been has the tDNA inserted into the genome. Anyone see that? Yeah? More or less? You can ask. Well, we will see more in a second. So, yeah, so basically you got agrobacterium. This is a cell from the plant. This is an example of a cell culture by tobacco, okay? And you can just put agrobacterium in the cell culture. Agrobacterium will transfer by very complicated mechanisms that we still don't understand fully, the plasmid into the cell, and then from the cell, the tDNA will get transferred into the genome. And this transfer is actually random, okay? We will see more about that. But it just gets inserted into the plant genome randomly. You cannot tell where to go, okay? This cell culture, you can grow them in plates with MS media to actually produce calyces, and some of them will produce some plants or develop some plants or plantlets that they will actually be able to be transferred into soil. And then you can see if the transgenic plant is actually or where the tDNA insertion has been inserted. To be able to just do these culture cells and actually choose which one contains the tDNA inside the genomic cells is what you require this selectable marker, okay? Because if not, you will be growing hundreds of thousands of plants, and maybe you have only one. So with this, you actually clean up quite a lot. This is an example of one of these tDNA biotechnology examples of how biotechnology has mutated all these things. The key parts of a tDNA plasmid is to know where are the borders. So the tDNA border or left border is here, and the right border is there. So basically, the only things that they will be transferring to the plant will be these bits, okay? This other part of the plasmid, as you can see, has the bacteria selection gene, ampicillin, kanamycin, things like that. The original replication for E. coli, and a few other genes that they are required for agrobacterium.
Well, I'm just thinking for the audit method, you can, in lots of cases, you can use the same selectable marker for bacteria and plants.
Yeah.
Antibiotics can stop produced.
It's in the next slide. Sorry. I will come back to that, yeah. Fluoradipin is amazing. So, yeah, no, sorry. But yeah, keep the thought, okay? So you have the selection for the bacteria, and then in the tDNA, where you actually it gets inserted into the plant, or the part that you want to put, will require the plant selection gene, okay? And in this case, you're right. You could use kanamycin resistance for both, because it works in plants, okay? But if you want to have two different selections, you can actually use a herbicide resistance, like Basta resistance. That is quite important for this type of experiments, because you just have to basically fumigate with the herbicide and see which one survive and which one not. Obviously, you need promoters for these selection markers, and you can always, all these tDNA plasmids come with a multiple cloning site where you can put whatever you want to put. You want to put the sequence of a gene of another species or the same species, or whatever you actually want to obtain. We will see also RNAi, so even how to put the CRISPR-Cas system into the plant. So all this is quite handy. And that's it. Basically, this is the part that it will be transferred into the plant genome. And this is the fluoradipin system. So once you do one of these experiments that you want to say, okay, for example, for the complementation test, okay, we have a mutant phenotype and we want to make sure this gene that is mutated is the real gene that actually produces phenotype. So you take the mutant and you transform with tDNA, expressing the gene from the wild type. Yeah? So what you do is use this tDNA plasmid, cut in the restriction sites that they are inside the tDNA that is going to be transferred, and you amplify the gene of interest that you want to test and include it into the tDNA. Okay? So that will be that yellow part there. Basically, you pass this plasmid from E. coli to agrobacterium. Agrobacterium could be put into the plant cells, or in this method that is called flora dipping. Basically, you take the flowers from the plant and submerge them for 1 to 2 seconds into agrobacterium solution. And then you just let them dry, and agrobacterium will go inside the cells to produce the tDNA transformation. At the end of the day, you will have your plant chromosome sequence in green. You will have the left border, right border of your tDNA, and inserted in there, you will have the gene that you have chosen. And obviously, you will have the selection marker that it will help you for the next step. So once these plants produce seeds, you collect the seeds, and then you grow them in plates. So you can grow them with the selection marker. So it could be kanamycin resistance, or it could be herbicide resistance. And as you can see, the seedlings that they are white, they are dying because they are not resistant. The ones that they are green, they will survive because they are. Okay? And that's basically what we do in the lab most of the time when we are doing this type of analysis. Okay? Because one of the things is because we know the sequence of the tDNA, we can produce primers to sequence outside that region into the plants that they've been selected to have the tDNA insertion. So we will be able to sequence outwards the tDNA and know where the tDNA is inserted into the genome. Okay? And that screening, it will allow us to, or it has allowed us to do things like this. So this is the tDNA Express webpage from the Salk Institute, and they basically have done that in thousands of times. And they have managed to screen tDNA insertions, all these Salk pink things are tDNA insertions in different genomic regions. So because they had done the screening and they know where the tDNA has gone in all these individual lines, they are able to sell us to us. So once you need a tDNA insertion in one of the genes that you are interested, for example, let's say this one here, so you just go to the Salk line and check, okay, I'm interested in that Salk line that it has a tDNA insertion in the gene of my interest, so I can analyze. Okay? And normally are $4.5, although they charge you $8 for sending it, but it's quite a very good system to actually work. Okay? And because you know where the tDNA is inserted, and you know the sequence of the genome and the sequence of the tDNA, you can actually genotype by PCR every time in the lab. So it's a very good system to know if actually the tDNA is inserted or not, where they say. And this is basically what genotyping is. You know your genome sequence where the tDNA is inserted, so you just produce a primer before and after the insertion, and a primer into your tDNA, and you can actually know the wild type will amplify only the genomic band that is normally 1 kV, and the mutant will not be able to amplify tDNA insertions, so the tDNA sequence is around 9 kV, 10 kV, so the polymerase won't be able to amplify between the genomic primer LP and RP, so you won't have the band of 1 kV. But if you do a PCR with the BP, that is the tDNA specific primer with either LP or RP, you will be able to see a band. Okay? That it will be, normally we design it that is a little shorter. So you can actually do, the main thing is that you will know if you have a wild-type plant or you have a heterozygote, so one copy has a tDNA insertion, another copy hasn't. And if it's a homozygote where you don't have genomic part, but you have both of the two copies actually have the tDNA insertion into the gene of interest. Or actually, if you want to express something else, the tDNA will be expressing in that sequence. The problem is that the tDNA actually gets inserted randomly. So if you want to do an experiment like the complementation test, you have to make sure that the effect that you see is nothing to do where the tDNA has been inserted, but because you are expressing what you are expressing, okay? So you will have to analyze different lines and actually check where these different lines have been inserted. And this is, again, the selection marker was a problem for the GM controversy with quite a lot of products, and not knowing where these insertions can go. That's the other major controversy in GM, basically. So, as I said in the example, you can do a tDNA insertion and mutagenesis. You can actually put a sequence of genes that could produce a recombinant protein with GFP, mCherry, anything that you want to do. You could do that with different tags that you could actually pull down the protein. So this is basically biochemistry and cell biology that you have seen in any other organism. So we can do all these things in plants. Yeah? So basically we use different tDNA lines that you could actually insert your sequence of gene with GFP or with mCherry. So you just play around. So you just do normal biotechnology experiments. And then the tags could be fluorescent recombinant proteins, so they could be just tags that you can actually use specific antibodies like CIMIC or his tag to actually pull down that protein later on when you want. What would I do like to have a pull-down protein? Anyone? What kind of analysis could I do with that? If I'm interested in protein A, and I pull it down using one of these specific tags from the total amount of protein in the plant, what will I be looking into? Protein interactions, yeah. So you could actually build a network of what this protein is acting or interacting with, okay? You could actually just check location of the proteins. You can do quite a lot of different things. So you can do immunolocalization, or if you use a fluorescent tag, actually see in vivo where this protein is. The other technique that is also pioneering plants for the first time was RNAi or RNA interference. And you know all this, so I'm not going to go too much into that. But basically, it's based on what we call hairpin RNAs. That is just a sequence that has some complementary sequence on the same sequence that they will actually form a secondary structure like this. There is the massive machinery of the RNA that it will recognize it. It will break it in small interfering RNAs that actually will go to the RNA messenger of the actual gene and actually degraded it. So this is what I normally know that is a very good control of the translational repression or actually the amount of RNA messenger that is available at a specific time in a cell. Now, if we do the same kind of thing, we have tDNA plasmids where we can insert our hairpin-like sequence. So basically what we call is the sequence of the part of the gene that we want to actually trigger or for the RNA interference that we call sense. You do the complementary reverse sequence, that is the antisense, and normally you put the loop in between that it will produce this loop on the hairpin RNA that normally is an intron that actually just stays there. This will be degraded into small RNAs and will actually go to the RNA messenger of that gene that you want to trigger and actually, or target, and actually just reduce the amount that you have. There are lots of different vectors with different selection markers, different promoters. Some of them are inducible, so you could actually induce the expression of the RNAi, And actually, depending where and how much you put, you could have more or less knockdown. So that's quite really nice. And there are different clone technologies that, like in any other system. So they can produce knockout, so null mutants. But the beauty of RNAi is not just that, is that you have an average of some plants that actually they are nearly knockdowns. They are in between the wild type and the knockout and completely wild type like. So you have different lines with different effects that you could analyze. And this is very important because if a specific gene mutation is lethal, you could actually be able to analyze different effects on that line, okay? In the other hand, it's very important too for gene redundancy. Anyone can explain me gene redundancy?
You've got two genes in an organism that do pretty much the same thing. Like Arabidopsis has got 5 delta proteins.
So
you knock one down, there are 4 more left that do the same job, so you won't detect a phenotype.
Mm-hmm. So basically, plants, not only Arabidopsis, they have quite a lot of different copy numbers of specific genes. If they are important, plants normally get 2 or 3 copies for reasons that we don't understand too much. And normally, if you produce a mutant of one of them, the other copy can actually work perfectly right, and you don't have any phenotype. So the beauty of the RNAi, because these genes have similar sequence, is that we could use that similarity sequence to actually produce a hairpin that could affect both of them, and we could put the knockdown in both of them. So this is an example of an RNAi construct for a gene actually that is related to pathogen responses. And basically, it's a negative regulator, so in the wild type is stopping the expression of other things that actually will produce an effect when pathogens come. And if you knock it down, you have an effect as like a pathogen is interacting with the plant. In this case, you see the leaves basically get twisted compared to the wild type, and actually some of them are quite dwarf, so they are very small and starting to die actually. That is what is normally happen when there is a pathogen. They want just to get rid of that leaf that has been affected. This is another example where basically you have your wild type without no hairpin, and they were knocking down 2 or 3 different genes. So they use a hairpin, the sense and antisense, that they was conserved in between these 3 genes. And actually, you can see that in the wild type expressing the hairpin, they have the similar phenotype like the other one. So it's actually quite good for this kind of gene redundancy. So one of the things that we will relate it with the workshop is actually genome targeting, that is bringing a new completely horizon for plant genetics, because we will be able to target where we got the mutation. And so we will see more about that in the workshop. So any questions? Okay. So please read the article and come for the workshop. Well, we have first lectures and then the workshop. Have you signed the register? All of you? Anyone? No? Okay. So see you next week.